Hello, RDs and RDs to be. Welcome to the RD Exam Ready podcast. I'm Zach Caseberg, your tutor, coach, and guide along the path to passing the RD exam. My biggest passion is to help RDs and RDs to be just like you to know how to efficiently pass the CDR exam. If this sounds like you, then hey, you're in the right spot. This is going to be our third edition today of a biochem session, and we've just been focusing on a single macro at a time. We've done carbs, we've done protein. Today is now time to do biochem of fats. I've said it before, I'll say it again. You as an RD, you have a responsibility to know biochem and know about these macros. It's what's separating you from the run-of-the-mill nutritionist that's out there. It shows you know what you're talking about. It shows you understand MNT and the basics behind it. So it's very important that we get this down to the cellular level. You're not being asked to be a biochemist, but since we work with food, we must know about how our body uses that food all the way down to that cellular level. Let's peer in and let's really dissect the topic today. On the CDR exam, biochemistry is actually just focused on the basics, like I said. While it might not feel too basic to you when you're studying it, trust me, the exam is not looking more beyond the basics of what we need. That's the good news. What's most important on the exam is understanding the following: the inputs and outputs of each key process, where the process occurs in the body, what conditions trigger a different process to occur or another one. And if you use some tools to help you out, you can really go about learning these things instead of just relying on memory. Make sure you draw things out, write it onto note cards, make mnemonics if you'd like to, and tell a story with biochemistry and digestion as we go through these here. I want to warm up with the very basic facts of fats. So many things in your body have to do with fats. From the bilayers of your body to the cholesterol in your blood, fats are everywhere. Fats are defined based on the properties that they do and a bit about their structure here as well. Fatty acids are the simplest form of lipids, and they look like these long chain of carbons. On one end, if you can kind of see what I have here, we have this carboxyl group, and this carboxyl group is very hydrophilic. It likes water. And then we have all these carbon chains here too. Next to it, we have the methyl group. And the methyl group down here on the other side, that's going to be the hydrophobic group. What you notice about these free fatty acids is they're usually just in a chain, or I'm sorry, they're just kind of all chained up carbons in a line, is what we see. Now, that structure is determined by the level of saturation of that fatty acid. So, picture that fatty acid that I clumsily tried to bring onto the camera here. As you know, we have our saturated fats, which are our bad fats. They are saturated because they're saturated with carbons. And that is because saturated fats, they only contain single bonds between their carbons. This makes them very linear because there's no bend or change at all inside of them. The next one of a type of fat that you should know would be a mono unsaturated fatty acid. These fats, they have a double bond somewhere in the chain, and this causes a bending effect to occur with the fatty acid. So it's not straight, it bends. Polyunsaturated contains multiple double bonds, even up to 6 double bonds. And this makes it so that the molecule is unsaturated with the hydrogens, okay? That's why we call it unsaturated. Both the mono and the polyunsaturated fatty acids have that bend. So they're bent like a U or like a horseshoe. And this greatly contributes to the reason that these fats are prone to oxidation, why they're beneficial for our health, and why they're liquid at room temperature. These are all reasons, this level of unsaturation, that does this. When they're bent like a horseshoe, they're all on the same side. And what we call that, when it's on the same side of kind of the curve of that fatty acid, we call that a cis fatty acid, or same, when it's kind of oriented in that U with the 2 points pointing the same direction. Trans fats are another type of unsaturated fat that you've heard of. What we're doing with the trans fat is we're not making a U. we're more making like an S, or we're going to reorient one side of that unsaturated fatty acid and we're going to turn it and rotate it. So now we don't have a U, we have more of a linear structure while still being unsaturated. And this is what food companies loved about trans fats is they go, "Hey, we can get fats in here, we can make them trans, and we can make them I want to say much more stable to heat, to energy. They're a lot cheaper to do this, and you don't have to invest so much money in it because you're using a trans fatty acid." Luckily enough, trans fats are no longer allowed to be added in quantities to our food. They're still used very slightly, but in such small amounts that it doesn't build up to a nutrition information amount. Now, there's three fat molecules that we need to know for the RD exam. We talked about the fatty acids, but fatty acids are used to build bigger molecules. Firstly is triacylglycerols, aka triglycerides as you know them. Triglycerides are found in all the food we eat, but as well as it's being the form of how fats are stored in the body. 95% of the food you eat, or the fats you eat, excuse me, is in the form of triglycerides. The structure consists of three fatty acid tails that are connected to a glycerol head. The fatty acids, those tails that are there, can be saturated or unsaturated if they need to be. The next one we'll say is phospholipids. Phospholipids are phosphate containing lipids that form a structure of our cell membranes as well as surround all the organelles within your cells. Way down there, your nucleus, your mitochondria, they're completely covered in phospholipids, which is crazy to think about. They're also essential for components of lipoproteins, triglycerides, and other lipid molecules. Very good. Glycerol forms the backbone of phospholipids, and then they have an unsaturated and saturated fatty acid tails attached, and they have a polar head, which is hydrophilic. I don't feel you're going to need to know the exact structure of a phospholipid. Phospholipids are used in many things in food, such as lecithin as an emulsifier, and that's because of that phospholipid structure we just talked about. It has hydrophilic and hydrophobic ends, but phospholipids are very polar, more polar than that of other fats, and therefore they tend to attract water molecules while still being a fat. It's for this particular reason that they're used in egg lecithin and soy lecithin, as well as they make such great phospholipid layers or bilayers around our membranes because they can allow for things to pass through kind of through a water soluble membrane because they have that hydrophilic head that we're seeing there. So that's what's so helpful about phospholipids and why we eat them in the diet and use them in the body. They help everything get through and stay protected. Third one here will be sterols. Sterols are super unique. They're a four-ring steroid nucleus. They have one hydroxyl group, hence the name of sterol because it contains alcohol. Cholesterol is a sterol, and it also makes up many plasma membranes because it is quite polar. It also serves as the precursor for many other things in the body like bile acids, progesterone, cholecalciferol, estrogens, androgens, and other sex hormones. Now that we've gotten our background of the three main fats that we need to talk about, we are going to touch on their digestion. Predominantly, I want to stay with digestion just of triglycerides because that's what I think you're going to see most on the exam. There is some we could go into for sterols, for going into phospholipids, but a lot of it I feel is going to follow in the same path as triglycerides. So let's focus on the big guy here today. The story starts, of course, in the mouth with digestion. When we ingest fats in the mouth or any macronutrient, we get those salivary excretions that start to moisten the food in our teeth to chomp up the food through mechanical digestion. Related to fats in the mouth, there is one enzyme you might make a small note of. It's called lingual lipase. It's an enzyme in the mouth, lingual, that digests triglycerides. Problem is, this enzyme is only really present in infants, and that's typically why we don't talk about it in the basics of digestion and absorption. The fats after the mouth will go where? To the stomach. In the stomach, they're going to mix with hydrochloric acid, which is going to help just to help break things down and make them smaller. But they're also going to encounter gastric lipase, which is released from the chief cells in the stomach. Gastric from the stomach, lipase meaning lipid enzyme. Lipase, the enzyme, is primarily breaking the glycerol and those fatty acid tails, so it's breaking those apart, breaking those into separate molecules. This partially hydrolyzed lipid emulsion will then leave the stomach and enter the duodenum. When it goes into the duodenum, it's this acidic chyme, right? It's this acidic substance. And when it goes into the small intestine, it's going to be neutralized by pancreatic bicarbonate. Pancreatic bicarbonate is signaled to release from secretin, a hormone from the small intestine. We've talked about this a few times throughout our podcasts. Once the fats have entered the small intestine, it's now time that the hormone cholecystokinin comes into action. Cholecystokinin from the small intestine will first go to the gallbladder and it'll cause the gallbladder to contract. When it contracts, it releases bile salts into the small intestine. Bile salts, what they do is they really help to take these big fat globules and they help to emulsify and break them down into smaller fat globules. So essentially, we're taking a big molecule, breaking it down into very small droplets. Why this matters is this action is so important to help our enzymes to do their job. The enzymes suck at penetrating those fats. They need help in through the bile salts. They're actually being able to penetrate those fats because the surface area is much greater. CCK is also going to go to the pancreas to stimulate pancreatic lipase. This is going to break down our triglycerides and produce free fatty acids, monoglycerides, and diglycerides. They are now going to be in the form that is much more easily absorbed. Let's take a pause here. Picture it. In your small intestine, you now have some short and medium chain free fatty acids. The bile salts that we discussed earlier, they help to form what we now call micelles. So micelles are these big aggregate molecules, okay? And they collect a lot of the fats in the small intestine so that they can then be absorbed. Picture this micelle. It comes down to your small intestine, to your enterocyte, and what they do is they're small enough that they can tuck into that microvilli at the brush border. They then are almost like a bus. They're dropping off the lipids at the enterocyte so that they can get in. So now the lipids are transported across the membrane into the enterocyte, and then they're getting into kind of the lumen, or I should say more closer to the bloodstream is what we could say there. Once a lipid has entered the enterocyte, they're actually going to be repackaged, if you can believe that. It always blows my mind. So we've broken these down and now we're repackaging them. We need to repackage them in a form that can exist in the bloodstream. They're going to be reesterified in the enterocyte, and they're going to be formed into these large structures called chylomicrons. Chylomicrons are large structures that are used to transport lipids. So we took those free fatty acids, we reesterified and created them into triglycerides once again, and then we packed them inside a micelle. Does that make sense? Maybe. We'll see. These micelles that we talked about, they're mostly made up of triglycerides, cholesterol, phospholipids, sterols, those types of things. So for a quick review, we started with a triglyceride in the diet, and through digestion we broke them down with enzymes and bile acids to form this more free-floating fatty acid. The majority of those fatty acids, or a good amount of them, are going to be packed into these micelles. The micelle is the bus that takes it down to the enterocyte and drops it off. Now those fats are absorbed into the enterocyte, where they are then once again made into triglycerides, and then packed into something called a chylomicron. Okay, so to sum this up, fat digestion is really a bunch of breakdown, build up, repackage, breakdown, build up, repackage. Okay. Then we're going to see what the body does with these things from this state. So let's go into a bit of the biochem process of how these triglycerides are going to be treated in this case. We're going to talk about this in a fed versus fasted state to see if it helps us. In the fed state, we are going to absorb our fats as we discussed above. What's going to happen next is the fats are going to be transported by lipoproteins to the cells. There's also free fatty acids that may be bound up with albumin, a protein, and those can also get transported to the cells. And it could be going to the cells that need these components for phospholipids to make more bilayers. It could be cells that want to use fatty acids for energy right now. But when we're in this fed state, what I want you to imagine is we have plenty of glucose. We have plenty of everything. So likely you can assume of what's going to happen is these fats are going to get stored somewhere once they've been used up. This is going to happen in times where we have ample insulin. Remember, insulin is a fed hormone, and after I eat a meal, my insulin starts to rise. Insulin allows the fatty acids to enter the cell, and they do this through lipoprotein lipase. What happens is, remember that chylomicron takes the triglyceride and it's dropping it off at my fat cell. The triglyceride, it cannot pass into the fat cell in the form that it is in. So through lipoprotein lipase, we break down this triglyceride again so that the fatty acid can then enter the cell and the glycerol can enter the cell. It's amazing about how we just keep breaking this down in this case. So those fatty acids, they can enter that fat cell or a muscle cell via facilitated transport. There's some active transport that has been mentioned in the text, as well as some partial passive transport. Seemed like all of them were mentioned there. Once inside the cell, we'll go through lipogenesis to create a triglyceride once again, and then we'll store it in the fat cell. Lipogenesis is being done for other compounds here too. If it's somewhere where I have excess carbs or excess amino acids, then I can lay those down for storage and go through lipogenesis. But really what we're doing in that fat cell at that stage is we're breaking down all that stuff from the chylomicron, getting it into the fat cell, and then repackaging it into a triglyceride so it can stay stored inside the fat cell. Good. An alternate fat for fatty acids is they may be sent to the liver. Liver is also where it could end up too. In the liver, we can re-esterify these triglycerides with glycerol, and if we wanted to, we could just keep forming more triglycerides or more phospholipids in this case. So we could take our fatty acids and we could take them to the liver, and we could create other things too. So we could synthesize more triglycerides from other stuff if we wanted to. We could also do things like make some of our cholesterols that you may be curious about a little bit. We're not talking about cholesterols much today as far as the different types, but this is one outcome of where triglycerides could go. They could go to the liver, and the liver could use this to make different forms of cholesterol, which we can talk about maybe in another show. Transferring to the fasted state. In the fasted state, our body shifts the use of our energy stores. The body is going to utilize triglycerides for energy. Lipolysis, the breakdown of lipids, can occur in adipose tissue, can occur in other areas too. Lipolysis is going to take that stored triglyceride and it's going to break it into the fatty acid tails in the glycerol head. It's going to separate those things using an enzyme such as hormone-sensitive lipase. This is an enzyme that does lipolysis, for instance. The free fatty acids are transported to be used in the tissues, and glycerol would be secreted into circulation. To touch on glycerol, Glycerol could go to the liver and other tissues, and it could go through the process of gluconeogenesis. So that's one outcome for glycerol is that it can go to gluconeogenesis in a fasted state to create more glucose. This is awesome. Fatty acids will go to beta oxidation though. To touch on beta oxidation, it's a circular process, and you can picture the TCA cycle because it kind of looks very similar. As this fatty acid moves through this cycle, it releases FADH2 and NADH. You remember these from our biochem of carbs, I know it. FADH2 and NADH, these are both going to go to the electron transport chain to create ATP. At the end of the process, beta oxidation is actually going to yield acetyl-CoA as an output. And what are we going to do with acetyl-CoA? We're going to send it to the TCA cycle. And the TCA cycle will then continue to give us FADH2, NADHs, GDP, and all of this is going to factor in to create more ATP in the electron transport chain. When we've been fasted for a very long period, we actually need to rely on ketone bodies a bit more for energy, especially our brain, which can only really focus on carbs and ketones. And these ketones are going to come from fats. We have a ton of free fatty acids in circulation, and thus we have a lot of acetyl-CoA. The liver is going to take over at this stage, and it's going to take the excess free fatty acids and excess acetyl-CoA, and it's going to take it and oxidize it further to make ketone bodies. Ketone bodies are these acidic compounds that can be used for energy by the brain especially, and for the body. And you know this as ketosis here. Great, now that we've talked about the ins and outs of fats, we've done our review, let's apply this to some PQ. Remember, use the process of elimination to help pick the best answer here. Which of the following describes the process of breaking down fatty acids to produce acetyl-CoA for use in metabolism? Which of the following describes the process of breaking down fatty acids to produce acetyl-CoA for later use in metabolism? Okay, let's see about this one. A. Lipogenesis. B. Lipolysis. C. Lipase. Or D. Beta oxidation. Okay. Breaking down this answer. Okay. All right, so I'm looking for the process of breaking down fatty acids to produce acetyl-CoA. So I'm going to write A, B, C, and D, and I'm going to see what I've got here. For me, already, if we're breaking down fatty acids specifically to make acetyl-CoA, then I'm thinking we're in a fasted state. So we should be looking at things that are breaking down fats in this case. Let's look. For A, lipogenesis. Lipogenesis is not what I'm looking for. Lipogenesis will occur when I'm in a fed state, and it's when I'm storing more fat. This seems like I'm breaking down fats for use of in metabolism. It's not A. B, lipolysis. Lipolysis could be it, but lipolysis is kind of the process that we go through to break triglycerides there. And lipolysis is going to break triglycerides, but it's not specific to free fatty acids. I'm going to rule B out in this case. I don't think it's a good fit. Lipase is going to be the enzyme that, again, breaks down triglycerides. I'm going to put a question mark by it. D is beta oxidation, and I feel like this is my best fit because it's saying which of the following describes a process for breaking down fatty acids to produce acetyl-CoA. That fits best with beta oxidation to me because it's saying it's just fatty acids, and specifically the output will be acetyl-CoA. This is definitely my best answer for this one. Number 2, which of the following is false regarding triglycerides? Okay, which of the following is false regarding triglycerides? A, they contain nonpolar fatty acid tails and a polar glycerol head. B, they contain both saturated and unsaturated fatty acid tails. C, They can be easily absorbed into the enterocyte in any form. Or D, they can be digested by lipase enzymes. Okay, so I need to find the false statement here, RD to B. Let's see what we got. For A, they contain both nonpolar and polar glycerol head. They contain nonpolar fatty acid tails and a polar glycerol head. This feels like a true statement to me, and I need a false one. So I'm going to rule out answer A right off the bat. B, they contain both saturated and unsaturated fatty acid tails. Well, I know this is also a true statement, so I'm going to rule that one out too. C, they are easily absorbed into enterocytes in any form. This actually sounds like a false statement to me because I think I need to really almost like break the triglycerides down into smaller molecules to be able to absorb them into the enterocyte. I'm going to put a little check mark by C. Or D, they can be digested by lipase enzymes. This is also a true statement. So for which of the following is false regarding triglycerides for number 2, my best answer is going to be C. They are easily absorbed into enterocytes in any form. That is not a true statement. That will be a false statement. Number 3, describe the outcome of stored triglycerides in a fasted state. Describe the outcome of stored triglycerides in a fasted state. A, stored triglycerides will undergo lipogenesis to later be used for energy. Stored triglycerides will be mobilized by insulin and sent through metabolism. That's B. C is my cells drop off triglycerides to adipocytes for storage. Or D, glycerol is sent for gluconeogenesis and fatty acids are sent for beta oxidation. Let's read this one more time. We're looking for the outcome of stored triglycerides in the fasted state. So to reword this, when I have stored fat, how is it going to move when I am fasted or starving or anything like that? For A, stored triglycerides will undergo lipogenesis to later be used for energy. This is not correct because this sounds like what I would be doing in the fed state, storing triglycerides. So I'm going to rule out answer A. B, stored triglycerides will be mobilized by insulin and sent through metabolism. Well, insulin is again a fed hormone, and I'm looking for a fasted state. This seems quite off to me because insulin doesn't seem like it would be in a fasted state. I'm going to rule that one out too. C, my cells drop off triglycerides to adipocytes for storage. That sounds kind of weird. I don't think my cells do that. I feel like chylomicrons do that. And even still, I think chylomicrons would do that in the fed state, not the fasted. A little questionable on that. Or D, glycerol is sent for gluconeogenesis and fatty acids are sent for beta oxidation. This sounds like something I would be doing in the fasted state. So for number 3, describe the outcome of stored triglycerides in the fasted state. D, glycerol is sent for gluconeogenesis and fatty acids are sent for beta oxidation would be my best answer for number 3 in that case. That's a wrap for today's lesson, students. I hope you enjoyed watching or listening to today's show. Remember, passing this exam is a journey. We all need assistance along the way. Do me a favor, spread the word, tell your other studying RDs to be, recommend the show, rate, review, comment, or let me know how you think things are going. And remember, as always, happy studying, RD to be.